Welcome to this presentation on circular respiration, focusing on glycolysis, the Krebs cycle, and the electron transport chain. So, the circular respiration is a fundamental metabolic process that occurs in the cell of all living organisms. It converts biochemical energy from nutrients into androstenedione triphosphorate or ATP and release waste product. This process is divided into three main stages: glycolysis, the Krebs cycle, and electron transport chain. Each stage plays a critical role in effectively production of ATP, which is essential for various biological functions. Understanding those processes help us comprehend how cells harness and utilize energy. So, today's presentation will cover the following topics. Number 1 is the introduction, which I did. Number 2 is the glycolysis, wherein the reactant and products, the K enzyme and the step we're gonna be talk about. And number 3 is the Krebs cycle, where we gonna tackle the function and the key points of Krebs cycle. And lastly, the electron transport chain, which is we discuss the process and also the key outcomes. Okay, now let's have an overview on glycolysis. So, as we know, glycolysis is a causal metabolic pathway that breaks down glucose into pyruvate, yielding energy and other products. So, this process occurs in the cytoplasm of both plants and animal cells and involves a series of 10 enzyme catalyst reactions. Now, let's talk about the reactant of glycolysis. So, glycolysis are glucose. So, there's 2 ATP molecules and 2 electron carrier, which is the NAD+. And the products are 2 pyruvate molecules, which has 4 ATP molecules with a net gain of 2 ATP and 2 NADP molecules and 2 water molecules. So, next is the K enzyme in the step of glycolysis. So, let's look at some K enzyme and step in glycolysis. So, one is the hexokinase, the first enzyme in the glycolytic pathway, catalyzing the phosphorylation of glucose by ATP to form glycose-6-phosphate. So, hexokinase 2 is a fundamental insulin-sensitive tissue such as the heart, skeletal muscle, and adipose tissue. Next is the phosphofructokinase. So, it's a major regulatory enzyme in glycolysis. It's the catalyst of conversion of fructose-6-phosphate to fructose-1,6-biphosphate using the ATP. So, this step is the key control point of the pathway, influenced by various metabolic signals. And third is the aldolase. So, it splits fructose-1,6-biphosphate into 3 carbon enantiomer dioxyethylene phosphate and phosphate. So those are free energy released during glycolysis are typically used to form high energy molecules like ATP and NADH. The phosphate produced can then enter the mitochondria to fuel the citric acid cycles. So let's have an overview about the Krebs cycle. So Krebs cycle is also known as the citric acid cycle or TAC cycle. So this is a series of enzymatic reactions that occur in the mitochondria of cells. So these cycles play a central role in aerobic respiration, the process by which cells generate energy in the presence of oxygen. Okay, since we have the overview of Krebs cycles, now let's talk about the function and the key points in order to us to remember the Krebs cycle. So Krebs cycle has a 2 important functions. Number 1 is the production of intermediate compounds. So these intermediates are essential for the synthesis of various substances, including amino acid and fatty acids. Number 2 is the formation of ATP. So the cycle generates large quantity of ATP, the primary energy currency of the cell, through oxidative phosphorylation. Now, in order to remember the Krebs cycle, here's the key points. So first is the cycle begin with the concentration of acetyl-CoA with a with oxalate to form citrate, and through a series of reactions, the citrate is converted back to the oxalated, producing NADH, FADH, and ATP in the process. And lastly, you need to remember that this cycle also releases carbon dioxide as a byproduct. Next is the electron transport chain. So let's have a quick overview of the electron transport chain, or ETC in short. So this is a series of redox reaction that takes place in the inner mitochondria membrane. So it's the final stage of circular respiration and critical components of oxidative phosphorylation, the process that produces the majority of ATP in aerobic organism. Okay, since we have the basic idea of electron transport chain, now let's talk about the process of it. So basically, the ETC is comprised by several protein complex and other molecules that transfer electrons from electron donors, which can be NADH and FADH2 to electrons acceptor, which is the oxygen. As the electrons move through the chain, protons are pumped across the mitochondrial membrane, creating a proton granite. The flow of the proton back into the microbial matrix through ATP synthesis drives the synthesis of ATP from ATP to N-garnet phosphate. So that's how it's worked, the electron transport chain. So before we end this topic now, so let's have the K of SCOM. So the K of SCOM here on the electron transport chain are: one is the production of water as oxygen is reduced, that is also remember. The next is the generation of the large amount of ATP, which is used to power various circular process. And the conclusion of my report is that circular respiration is a complex but essential process that enables cells to produce ATP effectively by understanding the stage of glycolysis, the Krebs cycle, and the electron transport chain. So we can appreciate how energy is harnessed and utilized within the cells. So that's all my report and understanding about the Krebs cycle, the glycolysis, and the ATP, I mean the electron transport chains. And I hope you understand something on my presentation. And thank you for watching.